So today we'll discuss one of the important topic of CSR net syllabus in Unit 3 DNA applications. Like every day, we'll discuss 5 to 6 questions from previous year question paper, and for that you have to regularly watch our channels and subscribe our channels, follow our channels, and to get that you'll receive all the previous year question paper almost explanation in our channel. Okay, now come into that part. The unit 3, DNA replication. Now first question of the U level CSR previous year question paper, like you have labeled DNA in a bacteria by growing the cells in a medium containing either N14 nitrogens or heavier isotope N14 or heavier isotope N15. That is a say like you have studied in the Meselson and Stahl experiment. Meselson and Stahl experiment basically based on the same base Meselson and Stahl experiment basically based on the two types of nitrogens are used, N14 nitrogens and N15 types of nitrogens. These are N15 types and these are N14 types. N15 types and N14 types. Now, clear? So, you have isolated pure DNA from these organisms. Now, subjected to cesium chloride density gradient centrifugation. So, where you have two DNA strands are there. Now you have one light. First of all, here two types of nitrogens are used. One is N14 type of nitrogen, N14, that is lighter isotope of nitrogens. Another is the N15. N15 is the heavier isotope of nitrogen, right? N14 for lighter nitrogen isotope and N15 is the heavier. And these are not radioactive but it's on based on the density, as N15 have a greater density and density on the based on cesium chloride density gradient centrifugation. Now, first is that the DNA, in the experiment, the bacteria first grown in the N15 medium. Bacteria first grown in N15 medium. These are grown in N15 medium, so all the strand will be N15, N15, right? So these will be N15, this also will be N15. So in that, if we get the in the after centrifugation, we will get the bands in it's as a heavier, so you will get the bands in the lower portion, right? Now, in the show that all the cells are now heavy form. Now these cells are transferred to a medium on N14 medium. Then we transfer into N14 medium. N14, suppose N14 is the lighter isotope. So I am giving the green color of the N for N14, right? Now N14, the all the new strand will be N14 added. So from starting with N15, N15, now the medium is changed and medium is transferred into N14 medium. So the it will be a combination of two, because in first generation one from one bacterium it will produce two bacteria. So for because you know the formula n equal to n0 into 2 to the power n. In case of bacteria, n equal to n0 into 2 to the power n. N0 into 2 to the power n number, 2 to the power n. So n is the number of generation, right? Number of generation is 1, n0 is the initial one bacteria. So after first generation it will produce two bacterium. So that's way. Now here the band, you are getting the band after external centrifugation density gradient. You observe the band is located in between N4, N14 and N15. So if it is a heavier isotope, the band will be coming up. So, and for N14, N15, you are getting band here, band getting in the middle portion and the in the middle, and that's is the is a heavier, and these are lighter part and these are heavier part, so you're getting band in between. So it's proved that this experiment actually based on Meselson and Stahl semi-conservative mode of DNA replication. So answer will be your the DNA replication is semi-conservative mode. So we know that DNA replication occurs in three modes, that is conservative, semi-conservative and dispersive. In conservative, only newly in DNA strand, both will be new strand or both will be old strand. And in case of semi-conservative, one is new strand, another is the old strand. But in case of dispersive, the patch of old and new strand, right? So this experiment is shows the Meselson and Stahl experiment. That's semi-conservative mode of DNA replication. So answer will be real replication is semi-conservative in nature. So coming to the next question. Now, second question. So it's a semi-conservative mode of DNA replication. Now, all nucleotides, triphosphate, that is RNTPs, are present at approximately 10-fold higher concentration than deoxyribonucleotide or DNTPs. That is RNTPs present in 10-fold concentration than DNTPs. But they are incorporated into DNA at a rate that is more than 1,000-fold lower than DNTPs. So why this is how this is happened? Because RNTP's concentration is high, but during DNA replication, how DNA polymerase differentiate between RNTPs and DNTPs? Why DNA polymerase do not take RNTPs instead of taking DNTPs? Because you know during DNA replication, DNTPs actually deoxynucleotides are added instead of RNTPs. RNTPs are added in RNA, DNTPs are added in DNA. So now why this thing happens, now the so here this happened because the most of the there is a discrimination property, discrimination property, that is one is that you in case of a DNA polymerase have a distinguishing active in their polymerase site. There is a they can distinguish between the DNTPs and RNTPs, right? So these are RNTPs. See that RNTPs here the 3' prime. These are, and you know that primer is required, and primer provide the 3' wedge or hydroxyl group. 3' hydroxyl group react with the alpha phosphate of the 5' 5' phosphate of the adding complementary nucleotides, like these are alpha phosphate. Now here is that in the here some discriminating amino acids are there which can discriminate between this OH group in case of discriminate between DNTPs and RNTPs, right? This discriminating amino acid discriminate between the DNTPs and RNTPs. In RNTPs, there is a 3' OH group is present. This due to this 3' OH 3' OH group is present in both DNTPs and RNTPs. But in case of RNTPs, there is another extra OH present, that is 2' OH, because in deoxyribose there is a OH group absent in 2' position. But in case of RNA, 2' OH present. Due to this 2' OH, there is a steric hindrance occurs in the and this steric hindrance steric hindrance is detected by the some discriminating amino acid present in that. These discriminating amino acids which um, prevent the incorporation of RNTPs, though they are present in higher concentration. Now see that options. Options actually said, now the option actually says that, so although ribonucleotide, so first option says that, third option first we see DNA polymerase efficiently discriminate. DNA polymerase can efficiently discriminate between RNTPs and DNTPs because its nucleotide binding pocket cannot accommodate a 2' OH on the incoming nucleotide, right? This will be the correct answer because the It's a option 3. There is a efficiently discriminate between RNTPs and DNTPs. These are DNA binding pocket because RNTPs has 2' OH. This 2' OH creates a steric hindrance, right? And in DNTPs, the 2' OH absent. For that, there is no steric hindrance. That's why DNTPs and DNTPs are taken up. So DNA polymerase can discriminate between RNTPs and DNTPs by that way. Now, next question. DNA polymerase also have also able to discriminate between correct and incorrect bases, right? If it is a like there is a if it is a like there is wrong bases come here, like these are 3' OH group and these are 5' phosphate group, right? Now, these are in case of here, see that there is the incorrect base and there is no interaction with the alpha phosphate here. So DNA polymer is also able to discriminate between correct bases and incorrect base pairing, right? So now, now coming to the question number 3. Eukaryotic polymerase alpha tightly associate primase activity but moderate processivity. DNA polymerase epsilon and delta highly processive but lack primase activity, right? You all know that DNA polymerase alpha have both primase as well as polymerase activity, right? DNA polymerase alpha. Now, DNA polymerase alpha have low processivity. Say DNA polymerase alpha have low processivity. DNA polymerase. So there is a in eukaryotic DNA replication. Eukaryotic DNA replication in there is a switching occurs. The switching is called polymerase switching. Alpha alpha first adding the primers and created some initiator part of the DNA, and then it is replaced because there is less processivity of the polymerase alpha, and then the higher processive enzyme, that is epsilon and delta, come. So primers for both the strands are synthesized by the DNA polymerase alpha, followed by polymerase switching. Why polymerase switching occurs? Because of that high processivity of the epsilon and delta. And in Pol alpha is a low processive enzyme and also lack proofreading activity. So this and delta and epsilons have a it's a highly processive enzymes and it's have proofreading activity. So that's why your answer option will be option 4. Primers for both strands are synthesized by DNA polymerase alpha, followed by polymerase switching epsilon and delta. And in case of if alpha, polymerase alpha is a primase as well as polymerase, but polymerase delta synthesize both lagging and leading strand. Polymerase delta able to synthesize both lagging and leading strand, right? And always, so option 2, see that option 2, the replication is primer independent process. Replication, you get primer is always required. Primer without primers, there will not be the synthesis of DNA synthesis will not be occurs, and DNA polymer is never able to initiate a bare or a without primer, and DNA replication cannot be initiated. So leading and lagging strands synthesized by DNA polymerase alpha maintain the fidelity. This also not be correct. Pol alpha not both strand. So entire leading and lagging strand is also not correct. And only lagging strand synthesize need primer. Now both strand are required primer. So answer will be option 4. So next question. Question number 4. So these are answer 4. So now question number 4. For Escherichia coli chromosomal DNA replication, which of the following statement is true? Say bacterial replication, that is prokaryotic replication. It is asking. So prokaryotic replication, for option first, see that option. First one is in case of prokaryotes, different enzymes are required, like polymerase 1, polymerase DNA polymerase 1, polymerase 2, polymerase 3, DNA polymerase 3, DNA helicase, DNA guidase, right? Um, so, but primase enzymes, so different enzymes are there. So now the question option one, see that DNA polymerase 1 is the main polymerase required for DNA replication. No. DNA polymerase 3 is the main DNA replicating enzyme for prokaryotic DNA replication. So option 1 is not correct. DNA polymerase 1 is thought to identified originally by Kornberg as the one responsible for replication is not important for DNA replication first. So this is also not correct. So requirement of DNA polymerase 1 is in the context of removal of primer needed for DNA synthesis and then fill in the same with the DNA equivalent. Yes, DNA polymerase 1, we know that DNA polymerase 1 is the function of primer removal and gap filling. Primer removal and gap filling is done by DNA polymerase 1. Primer removal and gap filling are done by DNA polymerase 1. So we got the correct answer here. So its answer will be option 3. Option 3 is the correct answer. So also we'll see the option 4. DNA polymerase 1 is the primary enzyme for error-prone DNA synthesis in SOS response. SOS is the emergency response, so it's not be correct. So SS response, DNA polymerase 1 is not involved. So that's a um, answer will be option 3. So next question. So question number 5. So following a certain statement related to eukaryotic DNA replication. So you know that eukaryotic DNA replication and actually based on in eukaryotic DNA replication, there is multiple origin of replication sites are present. In case of prokaryotes, only one origin of replication site present, right? So first we'll see the option. So see that what D is present in every almost three options. So first we'll see the D. D is correct or not. So D is the says that the elongation rate of elongation of different DNA chain during genome replication varies drastically. The rate of elongation all of different DNA chains during DNA replication is not varied, so it's not be correct. Say so you can eliminate the option D from here, D from here, D from here. So, but you can see that option C. You know that pulse chase experimental technique. Pulse chase technique is used for to level the DNA replication. Yes, pulse chase leveling when during pulse phase actually radioactive alternate between the radioactive element and non-tagged element, non-tagged or unlabeled element, right? At during radioactive element is called pulse, and the during chase time these are unlabeled. It's a shift to the unlabeled compound elements, and that's from that pulse is explained. Okazaki fragment first discovered, right? That's these are correct. And during embryonic development, embryos undergo rapid cell division and origins are uniformly active. This is also correct. Genomes of multicellular animals contain many potential origin of replication. This is also correct. So answer will be option A. So next question. We'll see that one. Almost we have discussed. So now next is question number. Okay, so almost we have finished. So Now, I hope this will make clear.